name's Angela Panoskaltsis-Mortari. I'm a professor in the Department of Pediatrics and Medicine here at the University of Minnesota. But I'm not a physician, so I don't see patients. I'm actually a researcher, but I'm in a clinical department. So that's a little bit different, especially back when I was training. That typically didn't happen, but we see it a lot more now. So a little bit about me. I was told to say something about me. I used to be a dancer. So I've trained in ballet. I have a performer's grade in ballet. And I was Greek Canadian. My parents were immigrants from Greece. So when I first went to school, I didn't know any English. But I was very involved in the Greek community, and that's still a big part of my life. So I went to Greek school. As you see there, I did ballet, and I was on the math team and French club. And my favorite classes in high school were math. So I was this dancer who loved math and chemistry, but I also loved biology, English, business, Latin, French. I liked a lot of things, and I did quite well in high school. My dream job was to be somehow involved with experimental surgery. I didn't know if that really meant that I had to go to med school. Initially, I thought that that's what that meant, that I had to go to med school, but what I really loved, especially in high school, was doing all the labs in all my chemistry and biology and physics classes. I really liked doing the lab work and the benchtop science. The first job I ever had was working in a cheese shop, so I know a lot about cheese. Undergrad, I went to University of Western Ontario in London, Canada. I did a degree there in biochemistry. My favorite course was in enzyme kinetics, and I also loved doing the honors capstone project, which was basically, you do a lot of research and you write a paper about it. I was involved with Greek folk dancing. I had my own Greek folk dance group there. I taught at Greek school. I was on a radio station at the university. So I did a lot of things extracurricularly as well. I did a PhD in immunology and a master's degree at Western. I then moved to Greece. But the university at University of Alabama in Birmingham was wonderful. So it was a great place. I did a postdoc there in pathology. And after that, I came here to the university. I was recruited here to study the immune complications that happen after a bone marrow transplant. And so that's the division that I'm still in the pediatrics department and the bone marrow transplant, blood marrow transplant division. I became assistant professor 2 years later, back in 1995. And after that, I actually got a board certification in medical laboratory immunology, which is typically given to physicians, to MDs, but it enables me to direct a clinical testing lab. So as part of my job, it's kind of like what I call my hobby job, I also direct a reference lab where we do patient testing and clinical trials work. So currently, I'm a professor and vice chair for research. So not only am I not a physician, but I'm also in a leadership position in a clinical department. So that, to me, it had never occurred to me before that that could happen. And I'm also the director of the 3D bioprinting facility that I'm going to talk to you about a little bit. And I also direct the cytokine reference lab, which is part of that board certification thing that I got. So the kinds of work that I do are about trying to make tissues and organs. We're trying to bioengineer lungs using people's own stem cells. So I do a lot of advising of students. I have a lot of undergrads, graduate students, postdocs, fellows in my lab. I do a lot of research, teaching, and some administration. My other hobbies here is I'm actually the director and choreographer for the Greek Dancers of Minnesota here in the state, and I do a lot of volunteer work. So if you ever go to a Greek festival and you see folk dancing there, that's me. That's my group. So this is what we do in the lab. So this is one project where we're trying to make lungs. And what we do is we take pig lungs. We've done this with human lungs too, but they're so unpredictable to get. So we take pig lungs and we remove all the cells out of them, and they end up looking like this. And I don't know why you can't see the arrow, but I can see it. So the white set of looking lungs. Now you see how they still look like lungs, right? But there are no cells in there. And then what we do is we set it up. We have this all automated, so we have a bioreactor. So there are a lot of engineers in the lab that help us to develop these systems. Okay, we also need people who know how to write code, so software developing. And so we have this all engineered to be automated so that all of these fluids flow through these intact organs. And we've done lungs, kidneys, livers. And basically it works with detergents. So just like in your kitchen when you're washing dishes and there's all those oils and fats on your plate, they get dissolved by the detergent. In the same way, the detergents dissolve cells because the membrane of your cell is composed of a double layer of lipids, of fats. And so detergents can melt that away. So we remove all the cells. We create cells from what's called induced pluripotent stem cells. What those are is we are able to take, for example, a skin cell, reprogram it with 4 factors, and make it become like an embryonic stem cell. It's not embryonic, so we don't have that ethical issue. Okay? We take it from you after you're born or as an adult. We take a skin cell, reprogram it, make it become like an embryonic cell, and then we can make it become any cell type we want. We can grow up a lot of those cells and then recreate whatever organ it is people have an interest in doing. So here, for example, is a decellularized lung. So just to show you, see? And we can ventilate it. Look at that. There are no cells in there. Okay? It still looks like a set of lungs. In fact, I'll show it to you right here. Here's a decellularized lobe from a pig. Okay? And we've cut it in half, and you can come up later and look at it. But you can still see all the structure inside, all that sponge-like material, all those airways. Okay? The pulmonary artery. Isn't this amazing? This has been like this for probably 6 years. Okay? There are no cells in here. We can do this to your whole body. We've done this with an entire mouse. Still see that it's a mouse. So we could do it to your whole body. It would still look like you, but you'd look like one of the White Walkers from Game of Thrones. Okay? That's the only problem with it. There'd be no cells in you. So another approach that we use to make organs in my lab is 3D bioprinting. So what is that? So that's an automated type of layered manufacturing of biological material and live, with or without live cells. Okay, so don't confuse it with regular 3D printing. Okay, so you can 3D print whatever plastic object or metal object, right? Everybody knows about 3D printing. And this is also not about 3D printing with plastics, hard or soft plastics, to make a model of an organ, like to make a model of a heart and cut it open and look inside. That's plastic. That's used for surgical planning. That's not bioprinting. Yes, it's printing for a medical purpose, okay, to help you with planning your surgery or to explain to someone, here's what your heart defect looks like, for example. But that's not bioprinting, okay? Or like, for example, several months ago, I got a hip replacement, and the socket was 3D printed. Can you believe that? That's still not bioprinting, even though it was put inside me. Those are regular 3D prints. So bioprinting is about making a live tissue. And this is what Hollywood thought that would look like. So back in the top left corner, anybody recognize where that's from? Come on. Star Trek. Oh, come on, all you Trekkers out there. So that was the replicator, okay? What about this? Anybody recognize what movie this is from? Excellent. Fifth Element. Remember, they had found this one piece. They took this material, genetic material, the cellular material. They grew up tons of whatever biological material. And layer by layer, they showed their rendition of it, okay? This machine that reproduced this beautiful woman who ended up saving the world. Okay? That's what that movie was about. But, of course, that's not what's happening right now. So the purpose of bioprinting is the obvious, to make a functional tissue or organ, maybe to transplant it into a person or an animal. But importantly, it's also about studying disease. So making small three-dimensional models with human cells, okay, to study how cells behave, to study how you can maybe screen drugs using these small 3D models. Just about everything that's in your cell biology book and almost everything we know about how cells behave has been done on hard two-dimensional plastic. That is not at all normal, is it? All the cells in your body are in a 3D environment. They're juxtaposed to a lot of other cells, and it's a softer environment. It's not like hard 2D plastic. I'm telling you, they behave differently in a soft 3D environment. Another application is to actually print medicine, to print pills. So now people are exploring, and in fact, there is already one FDA-approved drug for epilepsy that's 3D printed. And so if you could print, so now people are thinking about printing multiple drugs into one pill. Okay, so imagine if you have like a whole list of pills you have to take. If you could get them all put into one pill, that would be much easier to take all your medication, right? And you probably wouldn't want to skip any. Also, you could, design the drug and design the pill, you know, according to the dose that is needed. So if it's for a small child, you wouldn't have to cut a pill in half. You could just, you know, put in the right dose and adjust the dosage as the child grows or as the disease changes. You can also print scaffolds to guide tissue growth. So here's basically what we do in my lab. So I have people who are good in software, kind of design stuff. So we do CAD modeling, okay, of what we want to print. We take some decellularized organ material. We make gels out of it. So a lot of this decellularized material, this matrix material, is actually composed of things like collagen. That's one of the most abundant proteins in your body, collagen and elastin. So we make gels, and then through extrusion, just like the way toothpaste comes out of a tube, we can then print whatever, you know, object we want. So here's, for an example, printing the scaffold of a nose. And you can see there's a blue light shining because that's what's curing the material and cross-linking it so it has some integrity. And this is what the bioprinting facility looks like. We have a lot of printers. We do a lot of training. We have all kinds of students come in to learn. We have different types of bioprinters as well. This is a laser-assisted bioprinter here. We have some that are homemade. And here's just some examples of some of the classes. In fact, Century College is now starting a program as sort of in conjunction with their additive manufacturing program, but it's actually on getting a certificate in bioprinting. So that has started at Century College. It's just starting this year in the spring. And here at the University of Minnesota, through the Biomedical Engineering Department, I teach a 3D bioprinting course. So the ways to get into this, you know, it's a lot of fun. The ways to get into it are multifaceted. You don't have, maybe you don't want to be actually printing or growing cells. Maybe you're interested in the patent law aspect of it. Or maybe you're interested in working with the FDA and going over the regulatory stuff. Or maybe you're an engineer who wants to make the sensors who are going to monitor these tissues in real time without destroying the tissue. Okay, so there's all these, you know, there's storage, transportation. So FedEx is interested in this field. You know, how we're going to transport the organs that are made. So there are lots of different ways to be involved in this kind of field. This is a picture of my lab, so it's a variety of people. And I'll just leave you with this. We already talked a little bit about Dr. Lillehei, who's considered the father of open heart surgery, what mankind can dream research and technology can achieve. Okay, so I'm happy to take questions. Thanks. Yeah, great question. So what we do is, so different ways to do it. If you want to keep the organ intact, what we do is we just connect the blood system, the blood vessels, to those tubes, okay? And if it's the lungs, we also put the materials, the fluids in and out of the airways too. So basically you just flow detergents through, and it takes, you know, a couple of days, and then all of the cell material comes out, and what you're left with is this extracellular matrix, collagen, elastin, and a bunch of other proteins that are just the scaffold, just like when you build a house and you put up a framework.
I know you can also, I think, make the scaffold like growing them on animals such as mice. How does that compare to the printing?
Right, very good. So sometimes you can make molds of the scaffolds or bioprint scaffolds and then put them into an animal so that they can be maintained in there, so that the body of the animal or the human is the bioreactor that keeps it alive and allows blood vessels to grow into it. Mm-hmm. You had a question?
I was wondering how much smaller you would need to, or like how small the current printers can print, and how much smaller you would need to get to like print capillaries.
Right, very good question. So the resolution right now that is reproducibly attainable with this extrusion type method is about 80 to 100 microns, which is not small enough for a capillary. There are the laser-assisted bioprinter. You can get down to the resolution of a single cell, which is about 10 microns, but you're still not quite down at the capillary level. So what a lot of people are doing is building main vessels that are larger in these 3D constructs. And then in particular what we're doing in my lab is then figuring out ways to get the blood vessels to sprout in certain directions. Or when you put it into an animal, yeah. You allow the blood vessels to grow. Yeah. It was, yes? Yeah, great question. It's exactly what we're doing, and in fact, our first publication on exactly that is coming out in a couple of months. So what we've done is we've been able to print cancer cells and a blood vessel, and then we've figured out a way to get the cancer to migrate in one direction and the blood vessels to sprout in the other direction so that they meet. And then we create this tumor that has a blood vessel supply. And then what we've been doing is, through the main vessel, putting in anti-cancer drugs and then seeing which ones kill the tumor and which ones don't. Yeah, great question. Yep. Any more? Yes?
So, kind of bouncing off of what she just said. So, like, have you made any breakthroughs with what kills tumors and what doesn't?
So at this point, what we're doing is seeing the drugs that we know work in certain animals or in humans that are in clinical trials to see that it's repeatable, those results in this model system, so that we know the model system is good. And then from there, we're then taking it to newer drugs. But what we are finding is that the cells grow differently, and that in this 3D environment with a lot of culturing of other cell types, because the tumor isn't composed of just cancer cells. It has some other cells in there too, other cell types. And in that context, these 3D models actually better mimic what happens in the body in the way that the cells grow slower, and also their response to the drug is slower. So the drug is not quite as effective as you would think from just exposing the cells just bare on hard, on a flat 2D surface, which is the way a lot of this work is done. So what we're hoping is that this type of work will kind of leap over a lot of the small animal model work that's currently done, because a lot of what's done in mice, all mouse experiments, doesn't translate to humans. About 95% of mouse research doesn't translate to humans. Okay, well, thank you so much, everyone, for coming.